Today we have Bythell joining us today. Thanks, Bythell, for accepting the invitation to speak to us. So Ben, he has a PhD in chemistry from Oregon State University in 2007. He was a postdoc at German Cancer German Cancer Research Center from 2008 to 2010. Then he joined the Mag Lab for a postdoc during the 2010-2013. After that, he got a position as assistant professor at University of Missouri in St. Louis. Now he's associate professor at the Ohio University. And today he's going to talk to us about the chemistry of useful destruction. So, Ben, please.
Thank you, Yuri, for the introduction. Thanks, everybody, for coming along. And thanks also to the people who have agreed to meet me. That's very much appreciated for your time. So, what is chemistry of useful destruction? Well, at least in my group, it means that we like to break things. And we like to understand why they broke the way that they did. Occasionally, this is laboratory equipment, usually not too, hopefully not too often. Mostly what we're interested in is gas phase ions, because we're interested in gas phase ion structure and fragmentation chemistry, in order that we can use those things to identify chemicals in a hopefully more useful way. So today I'm going to give you two quick example projects and talk a little bit about each. There's way more detail than I'll ever get into. The first one is to do with sugars. It's an NSF-funded project at the moment. And essentially, we're looking at a variety of sugars, and I'll get into some of the detail of why we might be doing that and what it's good for. Essentially, developing understanding of gas phase structure and then applying it. The second thing I want to talk about and touch upon briefly at the end is acidic metabolite identification. This is part of a collaborative NIH project with the quite optimistic universal metabolite tagging title for the whole thing. And it's concentrated, this part of it's concentrated on both discovery of new metabolites and improved quantification. Okay. So what do we do in my lab? My lab is broadly split into two areas. Mass spectrometry related techniques, some of which are listed here, either internally or through collaboration, which is part of the point of this trip. We do these things, and we're happy to do other things that you might have available. Or other people might have available. The second half of what my lab does, and maybe we're slightly better known for this, targeted computational modeling. So looking at gas phase ion structures, working out how and why things broke. And if you're from a more synthetic background, I think that this is a sort of gas phase retrosynthesis. Okay. So, mass spectrometry. The good news nowadays is that our equipment, compared to certainly when I was a graduate student, is tremendously good. In most measures that you can think of, we're superior. Sensitivity, mass accuracy, resolution, even in many cases reliability, but obviously that varies. And what that means is we're skilled at getting elemental composition information with reasonably high confidence. So that's all good. But it's only part of the story because the elemental composition alone doesn't give you the structure, and the structure is essentially where it's at, because that's where the biology or the industrial functionality of whatever molecule or group of molecules you're looking at comes from. So what we need is another approach. There are several. I'm going to talk about one of them. It's an old approach, but it's a very good one. So it's tandem mass spectrometry. And in this, we, and of course many others before, and us isolate and fragment precursor ion of interest, and then detect the resulting ions. And the good thing about this is the fragment mass to charge values that we detect are structurally diagnostic for whatever we started with. So in way of cartoon, here's the simplest way of doing this, that we use at least, collision-induced dissociation. It's an old experiment. Because it's effective, it's still probably the most widely used one. It's not very subtle, but it's pretty effective. After your MS, you're going to have multiple things. So I've represented this as the different circles on the left-hand side of the screen. We've made each of these a map. I'm going to ask you to imagine that the size of the circle is the relative mass to charge of these three ions. If we wish to then interrogate the middle mass to charge one, so the blue ion, we're then going to select that by electric or magnetic fields. In our case, it's always electric. Smash it into a gas with one or more collisions, depending on your particular instrument, transfer energy to it, enabling its dissociation into charged fragments and sometimes neutral pieces, we're going to detect the charged ones and then try and work out what we have from there. So very simple, conceptually at least. All right, sugars. So why investigate carbohydrates? Why would you care? Well, carbohydrates are vital components of essentially all biological systems, most definitely including us. If you do a stain for carbohydrates of cell material, you'll find that it all goes pink and purple. And that's because carbohydrates are absolutely everywhere. They're attached to everything, and they're on their own in the plasma and other areas of the cells, the body in general. So what this means is if you want to handle on biology and disease and things like that, you're going to need to know, be able to identify and ideally quantitate the particular sugars that are present in particular processes. And if you can't, you're going to have limitations in what you can subsequently do. So all right, my premise and what I pitched to get a faculty job in the first place was essentially this was an area that I thought we could hopefully bring some improvement to by understanding how things fall apart a bit. I'm now going to get to why these areas got quite so difficult. So first, a very quick primer on sugars. If you came to my house for breakfast, you would certainly be offered this sugar. It's my favorite sugar. It's lactose. It's in all milk products. But it's a sugar that only about 70% of the world's capable of digesting because of the enzyme necessary for it. On the left-hand side of the screen, as you look, you have a beta-galactose. It's galactose because the hydroxyl position 4 is up and axial instead of down and equatorial. This is linked by a glycosidic linkage, so an ether linkage to, on the right-hand side of the screen, carbon 4 of this glucose residue, which I have in red. And the reason that's glucose instead of galactose is because that 4 position is down and equatorial, excuse me. Okay, so that's what one looks like. Why is it so complicated? It doesn't look too bad in terms of a molecule. The reason it's so complicated is glucose has 20 isomers, one of which is galactose. As soon as you then start attaching these to one another, you get a multiple of that. And there's no, unlike something like peptides, which are linear, these don't have to be linear. So there's no guarantee that you should have a 1,4 sugar. You can have a 1,6 or a 2,3 and so on and so on and so forth. So immediately your number of possibilities is enormously larger. In fact, someone other than me did the calculation. If you came up with all the sugars that were possible, you'd have more than the mass of the universe, which obviously there can't actually be. Your other area of concern, of course, is I've labeled this as the anomeric sensor, but it's essentially the stereochemistry of the linkage. So at position 1 in each of your sugars, you can either be equatorial or axial, and that's going to affect the shape of the sugar that you've got thereafter and its potential function. Okay, so that's the problem, broadly speaking. We don't. The other thing associated with that is we don't have, because of that problem I just mentioned, libraries that are in any way, shape, or form comprehensive. We're getting better at them, absolutely, but we don't cover everything. In a mass spectrometer, when we break this, there's a general break the sugars. There's a general nomenclature. It's an ancient. And it's still around because it's really good, and it's this from Devon Costello. You can see here. So if we broke the glycosidic bond next to the galactose carbon 1, so the left-hand side of the screen, and that side kept the charge, we would end up with a B1 ion. If the right-hand side of the screen from that same cleavage kept the charge, we would end up with a Y1 ion. If we went the other side of the glycosidic oxygen, it would be C1 versus Z1. And if we did more complicated processes where we've broken more than one bond, we would end up with either an X ion or an A ion, depending on which end kept the charge, with the superscript indicating which positions within the sugar we've cleaved. Okay, so what's a spectrum of something like this look like? So here we have one, nothing too fancy. You can see we have our precursor ion 365, and we've got 4 major fragments. So typically as mass spectrometrists at this point, based on whatever ion was, so in this case lactose with the charging agent is a metal, what our job is going to be to infer the precursor ion structure based on these peaks. But it gets worse. So for carbohydrates, and then of course this is true of some other types of molecule too, you don't know which end of the molecule necessarily you've broken. So in principle, the B1 ion and the Z1 ion for lactose, well, definitely the B1 ion and the theoretical Z1 ion for lactose weigh exactly the same elemental composition. So there's no way of telling them apart in terms of straight mass. Same idea for Y1 and C1. So we have to deal with this difficulty, this uncertainty also. So how do we study gas phase ion structure? In my group, we use essentially anything we can get our hands on, but the main experiments are tandem mass spectrometry based. We vary the conditions of the energy conditions. So how much energy we put in, how fast we put the energy in, how much time we give the ions to react. So we're playing with reaction timescale as well as energy. We deliberately modify analytes to influence fragmentation. So we're doing series here. So we're looking at series of like molecules in order to see if there's patterns that would be usable in identification. And we do targeted isotopic labeling, deuterium, carbon-13, and less often, but sometimes oxygen-18 to guide the calculations part, which I'll get to in a second. The second part is, it's again targeted, and I'll say targeted quite a lot today, theoretical calculations to aid explanation of how and why each of the experimental spectra appear the way that it does. I can't emphasize enough that this is targeted because I've no interest whatsoever in calculating every sugar that ever existed. It would take forever, it would get very boring, and it would not be particularly productive. But we're looking for areas where we can help, areas we can provide some understanding that might be more generally useful. The long-term idea with this on the various projects is implement the findings to improve sequence and structural elucidation of whatever type of molecule we're looking at, ion we're looking at, excuse me. All right, calculations. First, a few gory details. I promise this is brief. We use either molecular dynamics or genetic algorithm simulations at the beginning. We've used various ways of doing this inside NAM for food and some other things. Most often with in-house parameters where we've either modified the code quite a bit or we've modified it very superficially just for our own convenience. And what this gets us initially is some bad news. And that's that these simulations straight away don't unfortunately produce numbers or barriers essentially that you'd want to stake a lot of your reputation on, certainly for small molecules. So you need some kind of subsequent quantum or density functional, essentially some kind of more realistic modeling thereafter if you're going to do this. So the good news is these approaches generate many structures. We run a program which analyzes and groups these into families based on dihedrals and various other things, from which the best candidates can be optimized. We use a hierarchy of levels, starting from the low level very fast, moving across to increasingly more realistic from left to right. This is one example, it's by no means the only example. We eliminate degenerate structures after each round, so you don't end up calculating 50 of the same thing the next time, that's just a waste of time. And reevaluate the families at each successively higher level of theory. We find for many things that this functional is a good compromise for hydrogen bonded systems. I am not religiously in favor of this, or that if someone else comes up with something that's just as good as faster, I'm going to change. The other good thing about the system is it's compatible with other models and rerunning additional structures if you're failing to explain the chemistry that you're trying to. All right, let's look at a couple of examples. The examples I'm going to show you today for the sugars are relatively small, deliberately because it's much easier to see things for a disaccharide than it is for something much larger. So I'm showing you two isomers here that actually also isomers of lactose. So these are glucose polymers. We've got cellobios on the top, which is beta glucose linked 1,4 to another glucose. And gentiobios on the bottom, which is a beta glucose linked 1,6 to another glucose. The only difference is in the position of the linkage. The residues are the same, the mass to charge is the same, the elemental composition is the same, charge state is the same. Okay. So, first experiment done the same day, using the same instrument. The only difference is basically we cleaned the instrument between the two runs so that we're looking at one to, this is ancient data, but illustrates the basic point. And what you see is that they aren't obviously the same. The stellar bias for this collision energy is doing a good deal more fragmentation relative to the Gentio bias. You're getting mostly B and Y ions with some A ion. The Gentio bias has additional peaks, and the B1 and Y1 peaks are much lower in abundance, although they're a ratio that's hard to see on this one. Is within uncertainties the same, and I'll come back to this. So broadly speaking, without making any big statistical claims or anything, what you're looking here is a difference of peak identities and potentially abundance both mattering, which is what essentially is used for sugar sequencing generally with the people that are doing this. I mentioned carbon-13 labeling, so I know these are B1 and Y1 and A ions rather than X ions. We also do some deuterium labeling experiments. The simplest way to do this, and I really like this experiment, particularly with new students, but it works for much more experienced students too, is you make your samples up, and of course these are standards. And you make your samples up, and you make them up in D2O or deuterated methanol, or a mixture of the two, and you leave them for a while, and what you get is the equilibrium shifts all of the hydroxyls, OHs to ODs, and then you can isolate a peak at 173 corresponding to 8 ODs from what's your isotopic distribution and fragment that. And you look at how that fragments and what are the mass to charge shifts resulting from that. Okay, now I'm not going to do all 5 peaks. I'm going to show you one example. I'm going to do this for the B1 because it's the easiest one to see. So when I initially did this, I'd done some calculations and I was doing this in parallel. It took ages for my instrument to arrive, so I committed the cardinal sin of calculating before I had experimental data. And based on the old literature, it should be the bottom left-hand corner, where you've got 4 deuterons labeled in red. So of course I did this deuterated experiment hoping it was that, because I had a barrier for this reaction and it being a good place to start. And of course I did that, and what I found was there was a 189 peak, but it was at about 1% abundance compared to the 188 peak. So 3 deuterons are far more prevalent than 4. Which means that essentially this is the primary mechanism is wrong. The bad news there is you've got at least 3 possibilities. You've got on the top left-hand corner this 1,2 anhydroglucose, and the top right corner this 1,6 anhydroglucose, and the bottom right corner this pretty angry looking to my eyes bitter-ionic oxycarbenium ion. So which is it? So we do some calculations. So first step we calculate here, I'm just going to show you a couple of cell bios minimum and get into how this works, how this happens. So the first one we have here is, this is the lowest energy structure at this level of theory for cell bios. You can see lots of nice hydrogen bonding, lots of charge solvation of the purple sodium cation by the hydroxyl and ether oxygens. Those are the ones in red. If you put at least 10 kilojoules per mole into this system, you can rotate the right-hand side of that essentially 180 degrees. And what that does is it brings up the primary hydroxyl group from the right-hand side residue to coordinate the sodium, and now you've got tri-coordinate instead of tetra-coordinate, but it's cost you energy to do this. Okay, so the global minimum GM is where we're beginning from. What about how we're breaking these things? So, I told you earlier that I tried this the original way from the literature from like 30 years ago and it didn't work, or at least the reaction didn't give us the product that was consistent with our labeling. So at this point we had to try some different things, and it turns out basically what you've got to do is get the sodium away from the glycosidic oxygen. If the sodium is next to the glycosidic oxygen, you get the wrong product. You extract a proton from carbon-2 and you end up with 189 with the labeling. So we get the sodium away from the glycosidic oxygen, we protonate the glycosidic oxygen fleetingly. This weakens the glycosidic bond, enabling nucleophilic attack, as in the top right-hand corner, of the carbon-6 hydroxyl oxygen into carbon-1 on the producing this. So you need at least 226 kilojoules per mole to this, and you produce this product complex of 1,6-anhydroglucose and glucose, and it's bridged by a sodium. Now, when I first heard about CID and stuff, I viewed it in my mind as a kind of fission process where you broke a covalent bond and things instantaneously shoot apart. Turns out that's not quite true. These product complexes exist for a short but finite time. And here, what you've got to realize is even if the complex is only around for a microsecond, on the life of a gas phase ion, that is an eternity because the vibration's on the order of a femtosecond. So you can do a billion vibrations in between, which means you can do some chemistry. And the main chemistry we do here is a competition for the sodium. So which of those is going to be more happy having the sodium? Turns out that's easy to calculate. It's approximately 182 kilojoules per mole, depending on whether you've got the alpha or the beta glucose. And the 1,6-anhydroglucose is lower, 167. The other thing to say there is you don't have to believe my numbers. Peter Armentrat's done this experimentally, and he gets the same answer as I do. Always a good sign in my experience. So, based on that, the Y-ion the glucose should keep the sodium more often than the 1,6-anhydroglucose. That's exactly what we see for the experiment. When we vary the nature of the left-hand residue, we vary this branching ratio and the numbers work. So you'd expect to see more Y1 than B1 here in general terms. All right, so I've told you quite a few things fairly quickly. What's this all come together? So glycosidic bond cleavage, the transition structure appears to be rate determining. So that's giving us how much the reaction is likely to occur. When we do these same reactions, which turn out to be the lowest energy for the two analytes, we get different barriers consistent with the amount of dissociation we were getting previously. And thus you would expect the B and Y ions to be much less prevalent for the Gen 2 bios than they were for the cell bios, which is true, yeah. And last but not least, we initially thought more data since supports us. We've got a means of crudely predicting branching ratio as well based on the sodium affinity of the products. And because I've shown you two beta disaccharides, question I'm always asked is, okay, well, what happens with the alpha ones? It's a good question. So the cellular bios and Gen 2 bios have cousins. They're maltose and isomaltose here on the right-hand screen. So the only difference is in the position of the glycosidic bonds. So here you can see they're down and axial, whereas before they were up and equatorial. Okay, so what happens in these situations? Turns out it's here slightly more complicated. We've got two competing reactions. So the first is this horrible strained epoxide forming mechanism. So that's this triangle. The TS means transition structure, so it's essentially your barrier, and this is shown for the alpha. You need about 223 kilojoules for this. It's strained, so it's entropically bad. I'll come back to this in a second. And from that, you can work out what you get. You get mostly Y ions because this is very bad at keeping. That's not the only way you can do this, though. And this was shown by the knee group too, listed in the bottom reference there. There's another way of doing this, and the other way of doing this is you make a 2-ketone. And this is a 1,2 H shift with concerted proton abstraction. Sorry, that's quite a mouthful. And essentially what you're doing is you're moving the red hydrogen at carbon 2 over to carbon 1. In fact, you're moving it as H minus. The transition structure for this is a little higher. However, this has a kind of a sneaky superpower, which is it's really entropically favorable. I can't really show you this well here, but it's much more spread out. So there's much more freedom for this to work. So what this means is, and we calculate rate constants over a range of energies to show this, that you would expect this ketone product, so this ketone reaction to beat out the strained 1,2 anhydro reaction in general. Okay, so this is how this one works. Now, whenever you have a conclusion to something where you're quite happy with it, you should always check it so that you don't fall too much into confirmation bias. We were fortunate to have a few conversations with Tom Rizzo's group at EPFL Lausanne, and eventually they taught me into doing some calculations on this monster. This is a cousin of the maltose I showed you. So this and others, these are this is beta cyclodextrin, so it's a cyclic poly alpha glucose, and they had fragments from this that they've then done cold ion spectroscopy on. So in principle, there's 4 possibilities from this that we could be generating. I'm going to show you these on this next slide here. There's the 2-ketone, which from the linear system we thought it should be. The 1,6-anhydro in B, the 1,2-anhydro in C, or if we were making a Z ion here, because we did not have the benefit of labeling, so you have to check both, we'd be making this different kind of alkene, we thought. We tried this. We did a lot of calculations, we did the spectroscopy, and by far the best fit for the data, and I mean like massively so, is this one here, and it's this 2-ketone. We've essentially got reasonably good matches for each of the bands, and even the doublet at relatively high energy. And the other thing I'd say is we've got data for the B3 and the B4, which we compare to Z3, Z4s, and all the other forms, and those also fit best with this ketone structure.
Is the bottom the predicted?
Yes.
Okay. And then do you know how long data were accumulated for in the experimental at top? Maybe like 30
off
seconds.
the top of my head, I don't know.
Okay. Yeah. Because I know Tom recently sped up the acquisition to 15, even as low as 7 seconds. I was just curious if it was along that time scale or not.
The problem is we did 2 different experiments that gave basically the same result, and I can't remember which convolution of the 2 we did.
Yeah.
The other thing to say is there's lots of cool front-end physics on this, none of which I'm going to talk about, some of which was developed here. I meant to say that earlier. So the mobility separation at the beginning. So far, this looks reasonably general. So that it looks like the mechanism is general. We're testing this with more systems and linear systems too, because if it works for the cyclic, but it doesn't work for the linear, that doesn't help you greatly. But it helps you if you know. It doesn't help you if you don't know. So a decent question at this point, and I've always asked this, so hence this slide, is does our approach work for larger systems? Yes, so far. We're building this up. It does take time. There's still a lot to do. Does the approach work for other fragments like cross-ring cleavages? Again, yes, so far. Sorry. This all appears to work. And then the next obvious one is do common modifications like N-acetylation matter? And again, yeah, you're changing the functional groups. You're changing the chemistry that's potentially available. Therefore, it makes sense that you should be able to change the way that you can break bonds and form ions. The good news about that is that you can we can identify chemistry specific to be it a GalNAc versus a GluCNAc versus a MANNAc, and then potentially offer a means of identification thereafter. All right, so in summary of this really quick talk on the sugar part, hopefully this very brief introduction has given you some idea of some of the things you can do with cationized carbohydrates chemistry. We've done anions, it seems to work there too, but there's a lot less work done. And how the structures, reactions, and intermediates might matter. We think our data provides evidence for prediction of the glycosidic and cross-ring cleavage barriers, essentially to predict how much of the reaction is likely to occur as a function of energy. And that the structural identities of your product dimer components enable prediction of your branching ratio based on the relative, in this case, sodium affinities. All right. Yuri talked me into talking about the Metabolites project, so here it is. A brief skirmish with metabolites. So I'm also on a metabolite identification and quantification project with Jim Edwards, mostly with Jim Edwards, Chris Arnett, and Gary Potty. I'll get to what everybody's doing, why we make a good team. And so the plan was to come up with ways to modify metabolites that were essentially hard to identify, let alone quantitate. And a good example of this, and the one I'm going to talk about, is things that are acidic or polyacidic. And there's lots of these within the body for various processes that we need to monitor. And so what we're doing here is a type of tagging. We've tried several different tags. I'm going to show you one example. And what we're trying to do is flip from something that's going to be singly charged very good at sticking to solvent, to salts, and generally giving uninformative fragmentation, to something that's more highly charged that hopefully breaks in a way that's involving carbon-carbon bonds and is a lot more useful to us. So this multiple form is called degeneracy. I'm sure there's people in the audience know much more about this than I do. So the idea is new structure-specific fragmentation chemistry at the end of this, or at least that's my part of the thought process. And so we want to get away from not structurally informative condensation reactions, because if your molecule loses water or carbon dioxide and it's an anion, that's not super surprising. It's also not very diagnostic, unfortunately. All right, so the examples I'm going to show you here are two things from Krebs or citric acid cycle, depending on what you want to call it. So I'm going to show you citrate versus isocitrate and then a couple of others. And the ratio of these is a function of time and body conditions changes. So they're important within metabolism. They're they're isomers of one another. Okay, so what can we do? So the tag design and synthesis we're trying to do three things. We're trying to have something that's basic so that it's going to attach a proton and ionize, thus be amenable to efficient ionization. We wanted to kill off the adduct problem we were having in the negative mode, and we wanted to be able to limit the proton mobility, because it's all very well having something basic, but if the first thing that happens is the proton moves from there into the middle to help you lose water, again, that's not super useful. So what we did was we put these benzene groups in between the amide functionalities and the charge bits. The idea is to keep the charges away from the carbons that are actually the backbone of your chemical that you're interested in. Okay. So that's what we did. Here's the spectra. This is Q-exactive spectra. And what you get from these two isomers is distinct spectra. Compounds are easy enough to identify. We get improved signal to noise, I believe it's 7 and 3-fold, if I remember this correctly, which in principle would enable you to quantitate more effectively. So what do any of these peaks actually mean, though, and what's actually going on? So in way of picture of here's the two, this is the lowest NH structure of each, and you can see that the charges, and I've drawn them on with these big positives to aid view, are essentially keeping these in wide elongated structures where they're spread out, keeping the charge away from one another. So that's where we're starting from in each case. What can we do in terms of the chemistry? Well, both of these precursor ions in this case are triply protonated, which gives us a nominal mass of 223. And what we found is you have essentially a common chemistry. And the common chemistry is abstraction of an alcohol proton to form either an aldehyde or a ketone, so form a carbon-oxygen double bond with concerted carbon-carbon bond cleavage somewhere else. So in some cases that involves spitting out one of the COs. So in the top left-hand corner, sorry, the citrate one, you see we're producing this 225 fragment, which is doubly charged, and a 219 fragment singly charged. It turns out 225 can fragment some more, and that's also diagnostic. On the bottom, we've got a 218 that's doubly charged and a 233 that's singly charged produced from these reactions. Okay, we got one to work, or two to work. Does this tagging method work for other acidic metabolites? Short answer is yes. We've got a ton of standards and we've got a mountain of real data we're working through, which I'm about to get to. So first I'll show you that we thought, well, okay, we're getting this alcohol proton being mobilized. If you have a cousin of citrate, which is malate here, you've got diacid instead of a triacid. In principle, you would expect the same kind of chemistry. And the answer is, yeah, you do. That's what you get. And in terms of showing chemistry, it's here. So again, we're abstracting an alcohol proton. We're once again making this 219 fragment. But now the complementary peak charge piece is 233. So that's this and other data that I'm not showing gives us confidence that this type of reaction mechanism is general for alcohol chemicals. So we're using this for essentially a lot of analysis of systems at present. So in general summary, we will, for the first part is what we think it's good for, the second part is what we're doing now. So Gary Paddy is on the project. One of the, other than being brilliant, but one of the many reasons he's on, one of the other reasons he's on the project is Gary has access to 2 buckets of yeast, which is really underselling it, because one of the buckets of yeast that Gary has, all the carbon is carbon-13. So what you do is you run the same experiment, same conditions that you treat the yeast in each case. You run it in parallel. Your high resolution data in each case should come off, your chromatography, the peaks should come off at the same time. So you should have essentially temporal information and then fragmentation information where both the precursor is going to shift by the mass of difference of 7 carbon-13s if you had a 7 carbon analyte. And the charge state, of course. And essentially we're using that combined with the tagging to look at this and search for new metabolites. The bad news with this is we've also been really good at thinking we've discovered new metabolites and then just discovered that what we were was much more sensitive. So we were getting things that would characterize another, good examples, glutathione. We were very excited for about 30 seconds. So that's ongoing at the moment. Chris Arnett is the synthetic chemist on the project. He's synthesizing a series of target compounds for this, which we're hoping to have done over the next 6 months or so that we can test them and hopefully we'll publish the next part of this essentially. Assuming we're going to.
How did those,
Sure. Sorry.
they probably chromatograph pretty well by reverse the derivatized.
So yeah, we could, I don't have it on here, but I can show you the 2 chromatograms.
Yeah, sure.
So you tighten them up, you get much narrower. So one of the problems with the citrate, isocitrate, I always get this wrong, is one of them does a ton of tailing. This narrows them. They're closer in time, but the peaks, the resolution of the chromatographic peaks is so much better that you're compressing all of your ion signal there, so it's helping you in that way too. And I mean, Jim is the chromatographer. So he and I have almost got opposite skills. I'm definitely a mass spec person that uses chromatography and he's the other way around. So it's very complementary. The other thing I'd say very generally is hopefully this quick introduction to a couple of things that we do in my project, in my group has been of interest to people. I'm always happy to discuss interesting problems in chemistry, mass spectrometry, or other things. And essentially all of the approaches that we talk about here are in principle at least amenable to other types of analyte. We've certainly done some over the years. Suggestions for potential problems or areas to investigate are pretty much always welcome. Lastly, well, almost lastly, I should thank the various people that have, foundations that have paid for the work that I've talked to you about today. My collaborators on spectroscopic things, Tom Rizzo, some of whose data I showed, Philip Netter, I have a bunch of data that I didn't show, my NIH collaborators, and Ohio University for hiring me in the first place. With that, I'd be happy to answer anybody's questions. I'm cognizant that I'm supposed to be done in bang on 40 minutes. I'm probably slightly early. So I'll happily answer questions should people have them.
All right. Thanks, Ben. You can sit down if you want, so we can show on camera.
If people can see my ugly face, I can come over here and apologies.
And then we can see.
I like to walk around when I talk.
Get that camera hooked up to like face corners.
Yeah, or maybe is that your personal laptop there?
Yeah.
Yeah. That's a good point though on the hybrid meeting thing.
Yeah, those things like
Yeah.
I mean, the whiteboard is cool.
Yeah.
Yeah, that's like the blind spot is the front of the room right now. So let's see if we have any questions from.
So I have a quick question. So you mentioned in your computational approaches, you're trying to do something to screen your initial structures, right? Mm-hmm. Can you explain a little bit about what you do there and how long does it take, for example?
Sure.
Like
so Basically, I think the sugars are a good example of this. So, I mean, I guess another good example would be peptides. But when you've got floppy polar molecules, what kills you, and you're nodding and that makes sense, what kills you is essentially you've got lots of possible minima that you can be beginning from.
I'm closing your presentation.
Okay.
Sorry, go ahead.
Sure. So what you need is a means of sampling those. So for the sugars, one of the tough parts you've got to be able to do is all the ring forms. Because some things they look exactly like, a chair form, and that's the lowest energy structure. And other things are nothing like that at all, and you don't know up front. So you either got to heat this up and cool it down to deform the things, using some kind of MD process, so we've done this in NAMD, or you use a genetic algorithm. So this Fafum thing, what it does is it systematically will change the ring shape and then run a low-level calculation that you can link to it. You can make it, if you've infinite resource, you can link it to a high-level calculation and cut out the steps. I don't have infinite computing power, so unfortunately that's not open to me. So what we're doing is generating lots at the beginning. Now, a significant percentage of these are not going to be that relevant to what's going on. But so we essentially start, that's why we start low and fast. So PM6 or HF321G are their representation of where the electrons are and how they interact is not very sophisticated, but they have the luxurious advantage of being really fast. So what they're good for is sifting out the terrible stuff. So you don't spend lots of time calculating something that's going to be 200 kilojoules per mole relative to your global minimum. Because there's a decent chance you could have broken your molecule at lower energy than that, so you don't want to be looking at it. So the family thing is essentially hydrogen bonding patterns and/or angles, which you can build in. You can do it multiple ways, one of which is using rotational constants out of the... We use Gaussian, again, we're open to using other calculation approaches that would generate the same thing. And basically telling them apart. And our competitors, the knee group, they do a very similar thing. Is that too much detail or not?
I mean, I'm just trying to understand, and I think it's,
sure.
Yeah.
I mean, yeah, ideally you'd calculate, if you had infinite computer resource, you wouldn't bother with the first steps. You'd start with the realistic model, put everything into that. You'd still do this to generate them, put everything into that and go. The problem is that's going to take a huge amount of time. And for this to be more generally useful, you want to be looking at as large and as interesting in terms of chemistry and in terms of general biological interest system that you can.
All right.
So it's absolutely pragmatism.
Yeah.
The full-on theoreticians would murder me for what's good enough for me. Sometimes we do have to go back in and use higher level stuff or single points and things like that too. Yeah.
Samantha's like, maybe.
I think we're using kind of a similar approach, right? Screening things out with a low level method and then kind of acknowledging that you have some ensemble at the end that's going to have these different rotations and things. So.
Oh no, absolutely. So every now and then I'll get a paper and someone will want to know why I didn't do coupled cluster with like a gigantic basis set. And if it was feasible, sure, I'd do it. If there was an advantage to it in terms of, if the answer was significantly better than the answer I'm getting, I'm not opposed to using something more time consuming. But at the moment, something like coupled cluster, for the size of systems that people are interested in, and the ones I showed you are relatively small compared to what people are interested in, it's just totally impractical.
Yeah. I have a question.
Sure.
Have you guys looked at other activation methods, like other than CID, or is that like of interest?
It's absolutely of interest. So we've done, with sugars, we've done some negative electron capture dissociation stuff. We've done some electron capture dissociation stuff, and even some electron detachment stuff. none of which is published. Some of that's us on our own for our own interest. Some of that's with Christina Hokinson of Michigan. So I'm absolutely open to trying other things.
If you have any questions from people that are online, you can guys also, if you want to chat.
I'm always has a hand up.
Go ahead, Mike. Are you muted?
Yeah.
Yeah, can you hear me now?
Yeah.
Yeah.
Yeah, thanks for the talk. I joined a little late. I don't know if I missed anything, but I think most of your labels are like the experimental part. Most of the labels are done in solution, right? Like in like when you're labeling your, yeah. Do you see any benefit of doing the labeling in the gas phase? Is there some unique things that may help solve some specific challenges? Yeah.
You can definitely do gas phase HD exchange on some analytes. In fact, back in the day when I did peptides, we did with Arpad Samaji and from Vicky Baisoki's group, we did hydrogen deuterium exchange of peptide fragments. In fact, that's actually in Jürgen Gross's book, which I couldn't believe. Like, I made that figure. Anyway, I digress.
Vicky is my PhD advisor, just as you know.
Yeah. So the problem with sugars, and Eliza Gallagher at Baylor has done a lot more on this than I have, is that if it's sodiate or if you've got a metal cation, is you've got to do the normal mechanisms, you've got to either have enough material colliding with it that you can do the exchange, or you've got, which means you need a higher pressure, or you have to accept that it's not going to be very efficient. So Eliza's done a bunch of stuff in source of this kind of thing. Another good person to look up if you're interested in this at all is Miklas Gutmann at University of Washington Medical School. His ion mobility, he basically used, his collision gas in the ion mobility is deuterated ammonia. And he does exchange that way because you've got something that's gonna abstract protons and then swap them out for deuterons. And he does this at varying collision energies, and he gets patterns that are distinctive for different types of sugars. So he and I have ongoing collaboration discussions about various things too. He's got some really nice work.
Cool. Yeah, I'm mostly trying to think about if there's any unique advantage of doing that in a gas phase. I mean, I was thinking maybe you're doing some like in situ sampling of some maybe tissue samples. It might be hard to do solution labeling.
Oh yeah, sure.
Right, something like that. If you can do it well in the gas phase, it might.
So Mike's approach, and maybe Eliza's, I'm not as familiar with Eliza's setup, but Mike's approach does definitely work for biological samples. I mean, he's at a medical school. If you're absolutely everything he does has to be applicable reasonably quickly in the clinic, otherwise he's being asked, why are you doing that? So I think the way to do it, maybe there's another way, a clever way to do it that no one's thought of yet, but I think his way would be the way to do this. So you basically he pressurizes the mobility in his, he's got a Synapse G2.
Mm-hmm. Yep.
I'm not sure how good it is for the lifetime of his Synapse G2, but you know, if the information's that good, yeah, he'll have to make some kind of decisions there. Thank you.
That brings me back to solid thoughts.
Somebody else. Oh no. Sorry.
I was thinking about Marcos trying to do isomeric separation ion mobility by changing the gas.
Sometimes you can introduce divalent metal cations and affect separation that way in the gas phase.
Yeah,
Especially works seemingly well for certain types of sugars.
Cool. Any more questions? Anyone? No? Okay. So I think we're Thank you again, Ben, for.
You're very welcome.
Yes.
Thanks everybody for coming.
Yeah.
Much appreciated.
Thank you everyone for joining. We appreciate it.
Yeah, it's like a real class. Thank you.
Yeah.